other funds. On behalf, it's
Yeah.
let me hand you the.
Wonderful.
Thank you.
Thanks, Daniela, and I'm extremely honored and very happy for this recognition and for this opportunity to speak and present. So thanks to SIAM, thanks to all SIAM officers and to all members, and thank you for being here. The purpose of today's talk is to give, let's say, general presentation of some of the things that I've been thinking about and working on for at least 3 decades. The goal is to recruit people to work with me and a lot of friends that I have that are passionate about what we do. As Daniela pointed out, I'm General Manager of Institut Roche. I'm also adjunct professor of mathematics. I'm actually at the Department of Automatic Control, but professor of mathematics. And then some images and diagrams have been created with different kinds of LLMs. Not very much, but I still point it out. So let's start with the scientific method, right? So the scientific method, the roots are ancient. So the old Greeks, Aristotle was working with induction, deduction, and trying to come up with ideas based on observation and then doing thought experiments and try to understand how nature worked. The thing that Galileo Galilei added in the 17th, 16th century was experiments. So really adding that you can have idealized settings and do experiments to verify your hypothesis. Still working also with thought experiments, etc. Then I would say that during the 17th, 18th, 19th century, we built the system we have today with kind of peer review and a community. The scientific journals started, and we started to share and build a real community of scientists. In the 20th century, I would say that statistical design came. So really trying to let's say that we started to measure more. So there were more data, and we had to dig deeper into the statistical design of experiments. I want to point out the FAIR data papers that came in 2016. This had been long coming because scientific research was more and more data-driven. So we needed to have principles to follow so that the scientific method would work. So, and FAIR was findable, accessible, interoperable, and reusable. Today, we have fleets of AI agents that help us to browse the literature, come up with hypotheses, and now we need new principles to secure that we follow the scientific method. And I will come back to this. But all of this, these are models. Like I had a linear line here. Of course, I mean, development in science is not linear. It's much more complex. So every time you put something like this up, it is a model, right? So, but what is a model? Well, being a pure mathematician by training and then starting to do applied mathematics like almost 4 decades ago, and then still entering biology 3 decades ago, when people told me, oh, I have a wonderful model for this disease, I was not thinking about this, right? But this is what, and it's just an illustration of the different communities And that when you speak, of course, it's easy to learn that a model can be an animal model, or it can be a set of differential equations, or it can be something that a lot of us in biomedicine today are excited about, organoid models. So more complex systems of different human cells that we can put together and make functional for weeks or even months to test more complex interactions in ex vivo systems. Or you can have some type of mathematical objects that I also will come back to. All of these things are models. And we think in models, and as G.E. Box pointed out, all models are wrong, but some are useful. I think it's from this paper, this quote. I couldn't find the exact quote, but I think this is the paper. And he also points out here that, which I found a little bit amusing, over-parameterization is often the mark of mediocrity. Right? This is what he says. And of course, he's thinking about things like von Neumann's quote that he said, give me 4 parameters and I can fit an elephant, give me 5 and I can make it wiggle its tail. Things like this, right? But of course, we know the modern LLMs, they have billions or even trillions of parameters. Mathematics is the language of human thought to some extent. If we want to make things precise in science, the mathematization of the subject is often a measure of how far we have gotten in terms of precise understanding. Il saggiatore, I'm a mathematician, so you have to excuse me, right, for somewhat overestimating the importance of our subject, right? That should be okay. This is a quote from Il Saggiatore from 1623, where Galileo points out that mathematics is the language of human thought. And nature is written in the language of mathematics. Another famous example is, of course, the paper by Wigner, The Unreasonable Effectiveness of Mathematics in the Natural Sciences. You've all read it. If you haven't, I mean, it's still very nice to read. And I think for us mathematicians, Wigner is probably more known for this paper in communications than for actually getting the Nobel Prize 3 years later. He, of course, in this essay talks about the quintessential system, the solar system, and how successful we have been in modeling it. I mean, Kepler came out with his laws in the beginning of the 17th century, built on the observations of Tycho Brahe. And of course, then Newton came. This, as you see, is done with an LLM. This illustration, you can find the traces. But the point here is that some aspects of idealized situations for apples and planets, they are governed by the same laws, the same mathematical models, and they are very, very simple. I just want to point out that these simple laws, as you all know, at least if you work in dynamical systems or in mathematics, is that these simple models can hold the seeds of complexity and chaotic behavior. So even the n-body problem, when n is bigger than 3, you can have extreme sensitiveness to initial conditions and get a lot of, let's say, behavior that is almost impossible from a practical standpoint to predict. The same thing will be true in biology. So what could we expect if we now want to model biology? So I first want to point out that, as usual, the subject and the knowledge is driven by a loop of technology innovation, of measurement innovation, leading to knowledge that then again goes in a kind of virtuous loop. So we learn how to observe things. We build a microscope to look at small things. This leads to new measurements that we can make, which leads to new knowledge, that leads to further innovation in terms of looking even more carefully at things. I here just selected a few highlights, again a linear thing. Crick and Watson, the discovery of the DNA double helix, 1953. Recombinant DNA and protein technologies, making it possible to cut and paste in DNA and in proteins. Monoclonal antibody technology, making it possible to create clonal expansion of B cells, producing exactly the same antibodies, so that we could design drugs based on monoclonal antibodies. The Human Genome Project, completed in 2003, back-to-back publications in Science and Nature, where we could read the 3.2, more or less, billion bases in one human genome. Then CRISPR gene editing technology, which made it even more easy to cut and paste in DNA, leading to gene and cell therapy, a revolution in gene and cell therapy. Alongside this, there has been a revolution in sequencing technologies, new ways to measure. When I started being interested in biomedicine, we could measure what was called at that time bulk RNA-seq, almost 30 years ago. So this was a mix of cells where we could extract RNA and look and count how many times we saw RNA from a specific gene, so getting measurements across the different genes. Now we can measure on the single cell level for hundreds of thousands or even millions of cells, knowing what the expression of the RNA is. On top of this, we can do protein measurements on the same cells. We can also locate them in 3D space to know the spatial structure. And I would say that this is expanding rapidly, and whatever we can dream of measuring, we will potentially measure. So this has led to an extremely rich and detailed set of biomedical data that drives new insights, but also holds the potential for unprecedented expansion and innovation. You know the Moore's law, of course. So computational capacity is growing exponentially. The volume of data is growing exponentially. The cost of storage is dropping exponentially. The data that is being created, captured, copied, and consumed is growing at a hilarious speed. If you look at the approximate rate now, more or less correct, it's 7 petabytes per second. That is how much data. So imagine that you print this. You go down to bits and print zeros and ones, and you print a normal book. And how thick would that book be? Let's say a normal-sized book. It would be twice the distance to the moon per second. So that's a lot of data, right? The PubMed database, which is where most biomedical publications are listed, grows by 3 articles per minute. So how do we synthesize this, and how do we extract relevant information from this wealth of data? Of course, we cannot do this, but AI can, right? Well, I mean, we can do it, of course, as humans. We have studied for years or sometimes decades, and we have a lot of knowledge and a lot of intuition. But to really follow the literature, extract what is relevant, and sort it for us, I think AI is a fantastic tool for us. So we have the digital infrastructure, we have the data, we have the compute, we have the AI, all of it developing exponentially fast. At the same time, in drug development, we see an opposite trend. We see actually an exponential decrease in efficacy in terms of how much does it cost to put a new drug on the market or give a new drug to patients. So the approximate cost to put a molecule on the market is around $3.5 billion US, and it takes 12 to 15 years. I think, and many together with me think across the community, across industry, that this can be really improved. This situation can be really improved. How do we do it? Well, I think there is room for mathematics here. There is room for a lot of different things, but I think mathematicians and the mathematical community can actually make a huge impact around this. So let me come back to this. The problem is maybe the traditional drug discovery and development pipeline. This has been very robust for good reasons. One of the things that I found extremely satisfying and good when I made the move from academia to industry myself was that it was so clear that ultimately whatever we come up with, we will put it into humans. So we better not be wrong. So this talk about reproducibility, scaling things up, was taken to another level, something that I, as an individual, as a person, really appreciated. But this is how it's done. So we have the basic research, clinical observations coming from the community, often coming from academic labs across the world, which leads to a potential for some target, some molecule, some pathway that we think we can drug. And then we start with therapeutic experiments. So really doing both computation in silico, but also in vitro. So really looking at human cell lines in glass dishes and doing experiments looking at how human cells in a very simplified manner will react to certain challenges. Then we move that into in vitro. So really trying to test on model organisms like zebrafish or mice. How does this really work in vivo setting? Ultimately, if everything looks good, we start to do safety testing in humans, so phase 1. We look at efficacy and dosing, phase 1, phase 2, and ultimately we take it to phase 3, where we look at a broader community of patients, and again look at safety, efficacy, etc. And a lot of these potential drug candidates, they fail at certain steps in this process. And I can't help thinking fairly often around, we test things in animal models. If it does not work there, it's very unlikely that we will continue to explore. And still, we know that most things that work in mice do not work in the same way in humans. So you can imagine that there are a lot of things that could potentially work in humans that we actually never test. I think the organoid models is one part of this, but I think an equally important part is mathematical modeling. So really using human data to try to come up with mechanistic and functional understanding. This is taken from an article, but it tries to point out some of the problems with the drug development process. Of course, there are a lot of drugs that have already been developed, so it will be harder to jump over the bar to get something that is really better than what is already out there. So that's one thing, right? The other thing is, of course, safety. We are getting more and more careful, I would say, in terms of safety signals. And this has to do also with the fact that we want to treat earlier and earlier disease. We really do not want to wait until I have a serious condition. We want to treat me before. And then, of course, if you have side effects, I'm not really happy to have a lot of side effects if I feel fine. It doesn't really seem to be that tempting. But I think the underlying fact of all this is that biology is very complex. It's very hard to reproduce, in detail reproduce a lot of research that is done in biomedicine. This is not because it's completely wrong, it's because the setting might be slightly different, and so you have slightly different results. Some of the basic underlying mechanistic and functional things might be true, but it's still very hard to reproduce exactly what has been done. And this, of course, doesn't go for all biomedical research. There are a lot of things that are reproducible, and we are getting better and better. And I think the FAIR paper that came a decade ago did a lot of good, and the FAIR kind of movement within the community has done a lot of good. But the underlying factor is that biology is complex, it's context dependent, and our understanding is slowly evolving. This comic strip is maybe not completely accurate in terms of how we see things, but I still find as a model, it of course wrong, but at least it's funny, right? A little bit funny, I find it. So models can also be highlighting in this way. But ultimately, if we want to put drugs into humans, we need to understand humans. And a lot of things can be learned from looking at human data. This is what we call reverse translation, or lab in the loop, where we observe patients, we go back to the lab to test hypotheses that we get from patient data, then we evaluate and go back. And so this is a virtuous loop where we include both ex vivo systems like organoids, in silico modeling, as well as human data. So there is no better model for human health and disease than human health and disease. Still, human organoids, as pointed out in this paper, has a lot going for it. And I think a lot of the work that we still currently do on animal models will be replaced by human organoid systems that actually replicate parts of the human biology much better than an animal model. But this is maybe a little bit where I think mathematicians can help, right? So we have a lot of knowledge in biochemical, conceptual models on how things work. This has to be translated into some kind of mechanistic, functional, mathematical models. Here, just illustrated with a very simple OD system, but you can imagine really complex things of reaction, diffusion, convection equations. Then we go for the data that we actually collect on humans, and we do in silico modeling, so really using statistical learning, machine learning, visualization to look for patterns. And then we have a loop here. And I think the glue between both the different models and between us as modelers, here I think generative AI plays a huge role. The holy grail, I would say, of at least for me and part of my team, is to understand and possibly be able to control the human immune system. So we are constantly exposed to pathogens, bacteria, viruses that normally do not live in us. There can also be internal perturbations, like our own cells that get mutations and go rogue, and we can have the formation of cancer. But we have a system that defends us, the immune system. And that is, of course, central to understand in autoimmune disease, cancer, infectious disease, neurodegenerative disease like Alzheimer, and most other diseases. I'm not trying to say that the immune system can be studied in isolation. It's coupled to every other thing, the metabolic system that goes on. But we have a focus on the immune system and how to model and maybe control it. These are a few points from Petter Brodin's paper. Peter has been working with me and with a lot of other people in the Milieu Intérieur project in Institut Pasteur. And this is a resource for the community. So if you're interested in results and also data on healthy humans and how they react, how their immune system works, this is a resource. It's not the only one. There are many similar projects around. But what we and others have discovered is that human immune systems are relatively stable within individuals. We look more or less the same over weeks or months or even years, but it's highly variable between us. And also the way we react to external pathogens or challenges is very variable. So this is something to bear in mind. So now we want to model a human. And then we have different hierarchical levels that are quite easy to recognize. They are organized on these levels, like molecules, cells that form into tissues, that form into organs, that form into organ complexes, that form into an individual, that are part of a population. And often we have measurements on patients on different levels here. It can be molecular level, cell level, organ level, and on the individual level. And we need to make sense out of these different measurements. So that is challenging. I think it's interesting to look at some of the length scales of life, just to have an idea in your head of what you're actually modeling. A virus is in length scale 100 nanometers. A bacteria is 10 times bigger, 1,000 times bigger in volume. A human cell is around 10 times bigger than a bacterial cell in length, 1,000 times bigger in volume, and so on. And we have a normal human is composed of around 30,000 billion human cells. We have as many bacterial cells in us under normal healthy conditions. And we have 10 times as many viruses under normal healthy conditions. So we are a bag of cells, and most of them are not most of the entities, most of the genetic information in our bodies is not what we call human. It's bacteria and viruses. But there are a lot of human cells, and they are important. And this is a very dynamical system. So around 4 million human cells die per second in an average human. Most of them are blood cells. Most of them are red blood cells, but a fair fraction of them are white blood cells, so part of the immune system. I think this book is a good resource for mathematicians learning biology. We like numbers. Here you get a lot of numbers. You can learn how many T-cell receptors actually sit on the surface of a T-cell, around 20, 30,000. And you can start to build those pictures in your head of the system that you're actually modeling. Let me go back to what I've been dedicating some time to exploring, namely understanding and controlling cancer. So this is an illustration of T cells that attack a tumor cell. And normally a tumor is actually erased by our immune system. So we have this challenge all the time, but our immune system defends us. This is at least partly the current understanding. We've been treating cancer for thousands of years by surgery, so cutting. Then, fairly recently, we came up with radiation, so burning. And then we came up with chemotherapy, so poisoning. And the Nobel Prize in 2018 was given to Allison and Honjo for the discovery of checkpoint inhibitors. So they discovered checkpoints on T cells, CTLA4 and PD-1, that we could actually block in order to take off the brake of the immune system so that it could do its work. And this has been a fantastic success for part of the patients. So in some cancers where we have highly mutated cancer cells, like lung cancer or melanoma, for instance, a substantial part of patients, let's say 30%, get life back. They are cured by immunotherapy. And this is very recent. And it's an example of how we go in and actually control the immune system. But we do not understand why, let's say, 2/3 of the patients do not respond to the treatment. And here I think that longitudinal measurements, modeling of the dynamics with the help of our friends, mathematicians and others, will be crucial to try to give benefit to more patient groups. A conceptual model that we've been working on in 2013, Ira Mellman and Dan Chen from Genentech published the cancer immunity cycle. 10 years later, they were joined by Tom Poles and Shannon Turley, and they wrote this follow-up. If you want to read just one paper currently on understanding, let's say, cancer immunology and how we work, I still think this is a really good paper to read. The cancer immunity cycle is a conceptual model that we use all the time to guide us. It should be read from 1 down to the left bottom. Release of cancer cell antigens. So these are cancer cells that die. When they die, the proteins in these cancer cells are chopped up into peptides that are picked up by cancer antigen-presenting cells, like dendritic cells, that are transported to the lymph nodes, where you have T cells that hang around. T cells have a lot of different receptors. Each T cell has its own particular receptor, thousands of them on the surface. If it recognizes its, let's say, key that goes into the lock, and then it clonally expands. So you get a clonal expansion of this specific T cell that recognized this specific antigen that came from the tumor cell. They clonally expand, then they traffic through blood to the tumor surrounding. They infiltrate the tumor, and the idea is that they recognize the cancer cells and they kill them. When cancers start to grow and you get a cancer, then some steps, one or several of the steps in this cancer immunity cycle is not working as it should. And therefore, it's a relevant conceptual model. We need to think about where can we intervene? Where can we help the cycle to get going again? The measurements we make are on genes. There are over 20,000 human genes. There are even more proteins because genes, they have different splicing, which leads to more proteins than genes. And we can measure hundreds of thousands or millions of cells for each human sample. So these are enormously big data sets. Still, every measurement you can plot into a very, very high dimensional space, and you can start to think about how does this space look like? Based on the measurements I'm currently have access to, how does this space look like? How can things move in this space? Of course, even without longitudinal data, you can still make things out because we are not that different between humans. So we can be in different stages of the same disease progression. We can look at other things like differentiation of cells, and they will be in different stages of differentiation, going from immature, naive cells to highly differentiated cells doing their job, etc. So we can learn things. And when we look at this immunity state space, there will be certain domains where we can find people that are healthy, and certain domains where we can find people that are less healthy. This is what we hope to be able to do. So this is what I call the immunity state space. So this is just an illustration. I see a matrix here. It's a small, small part of a very, very huge matrix. So 20,000 rows and 132 columns in this particular data set. But when you have a huge matrix like this, how do you extract information? Of course, we know that there are noise, there are artifacts. It's often fewer samples than variables, really hard. And we have an unlimited freedom in selecting what we want similarity to be, right? So we can have Euclidean distance or graph-based distances or kernel-based distances, you name it. Whatever we come up with, we can try it out, right? And then often, I mean, we humans, we cannot walk around in 100,000 dimensional space. We want to understand things in a reduced space. And the reduction can, of course, be done by generative AI, or we can, rely on more traditional mathematical modeling to do the dimension reduction. Both things are good, and both should be tried. Let me present actually one of the data sets that brought me into the field 25 years ago or so. So this I was presented with a very similar data set, over 100 kids suffering from ALL, so acute lymphoblastic leukemia. This is the most common cancer in children. This was bone marrow aspirates, so you extract a little bit of the liquid in the bone marrow, and this liquid contains a lot of blasts. So these are immature immune cells that are trapped in an immature state. So the particularity of this cancer is that it attacks actually the soldiers that should defend us against the cancer, right? It attacks the immune system. So what do you do? I mean, being a classically trained mathematician, the first thing I tried was classical principal component analysis. So I fed it into PCA. And a PCA, for those that are not mathematicians, it's basically, I mean, let's say that we want to study an object in 3D, like a hand or a pencil or something, and we live in 2D, then we can shine a light on this pencil, let's say, and look at the shadow. And depending on how we position the pencil, the shadow will change. If I position it so that it's a point, that is not very informative. If I position it so I actually see the shadow of the pencil, it's much more informative. That is because I maximize the variance in the projection. For a mathematician, it's not hard to do something go from 3D to 2D to 20,000 dimensional things, and then whatever we like to do, right? And this is what we do in PCA. It's color-coded here according to the clinician's diagnosis of the patient. So every point you see here is a kid, and they are color-coded according to which particular genetic aberration they happen to have. But this is not using this particular mRNA expression data at all. It's based on the clinician's evaluation. You don't see much. Actually, I don't see any patterns at all. But this is because there is a lot of noise. So most genes are just doing what they normally do. They just contribute noise, and you don't really see the signal. There's still a lot of hope here. If you if you look at the first principal component, it contains 21% of the variance. That is a lot for something sitting in 20,000 dimensions. So there is a signal here. Mathematicians like to look at scree plots, for instance, that actually maps out how much of the variance you capture in each principal component. So how much of the information each principal component contains. Now, if we start to filter away variables, this scree plot will change. And how do we, in an objective way, throw away variables to get information? So this is a paper that we wrote in 2011. Very, very simple idea. You look at the fraction of the variance you capture compared to all the variance, and then you look at the difference of what you actually have in your data and what you would get if your data was random, but with the same structure as the data, as your real data. And then you try to optimize this difference, right? By filtering away variables. So it's an objective criteria for filtering away variables. And the interesting thing is that when we tried this on a lot of real data, we saw clear optima. Like in the picture here coming from a specific data set. But it's just an illustration of what we saw again and again and again. There are clear optima in this, which I find extremely interesting. So I did this on 3D. And so now I optimized over projection score, and this is what came out. And as you see, I mean, we can use this data. At least there's a fair chance that we could use this data to classify patients. ALL turned out to be an easy case, actually, because most of the cells that we got in the aspiration were indeed blasts. So they were cancer cells. And let's say we were lucky for this, and it actually helped us to understand a little bit more of the structure in this data. So we did the same thing, exactly the same thing, but using graph distances instead. So we connected each patient to its nearest neighbors, constructed the graph, and looked at the graph distance, did multidimensional scaling. That is basically just, let me sheet a little bit and say this is like doing PCA, although it doesn't really work. But you see the result on the right here. And the interesting thing here is that the group, the light blue group other, splits up in two groups. And they actually turned out to have very different responses to the treatment that was used at that time. So this was a significant finding. It's not our finding. It's the finding of the previous paper. What I find interesting, and a lot of colleagues with me, is that we measure hundreds of thousands of things, millions of things, and still when we start looking for the signal, it's in low dimension, which is like a miracle. I think I have some explanations for this. Either, I mean, a very, very simple overview would be that either things are functionally connected and that reduces dimension, or they are statistically independent, and then you have concentration of measure. So things that you're interested in will concentrate highly around the mean for the specific function you're looking at. I think this is part of it, but I also think that a better and more profound explanation is that life operates in this way. It needs to be robust. Otherwise, I would not be able to stand here and talk to you, and you would not be able to listen and talk back to me, right? We would not be able to understand the same things, which is a miracle in mathematics. We all agree that this is mathematics, this is right, this is wrong. In biology, it's a little bit more gray, let's say. We can argue because it's contextual, it's harder to understand from many perspectives. Let me give, for the mathematicians here, a very quick introduction to how I think about the modeling. So measurements are not only measurements, right? They come with annotations. So annotations can be variable ID, sample ID, cell type, what did you do, did you drop it on the floor, anything that is relevant for this particular measurement. And the data structures is actually this triple of annotations, measurements, and the map in between. And I can start to model the measurements. For instance, I have maybe cell type as an annotation. I can take the mean for each gene or for each specific cell type. This gives me a new variable. So I have a new annotation and I have a new measurement. And in this way, I build out this annotation space and measurement space. And of course, the annotation space here is connected with all human knowledge. So if I say this is the cell type, when I started studying biology, there were a couple of hundred cell types. Now there are thousands of cell types. And we understand better and better that each cell is unique. And we need to decide where is it useful to try to say that they behave in the same way. They will respond in the same way. They are a cell type, right? This is evolving. And I think that the annotation space is really the realm of AI agents, and the measurement space is the realm of more classical math modeling. There are no sharp boundaries here, but I think about it in this way often. Looking for biomarkers is something we do all the time. You can think of A1 and A2 here in annotation space as responders and non-responders to treatment, for instance. We want to understand why. And then we are looking for, when we map this into measurement space, can we find structure that robustly separate these two classes? And here you can do anything you like. Graph geometry, kernel, feature space, sparse signals, whatever. And there's a lot of room. There are no gold standards. For example, when it comes to single cell analysis, RNA expression, together with spatial information, we don't have gold standards. We have a lot of techniques that are proposed, but I would say that at the moment we have no gold standards. Room for mathematicians to come in and help to come up with a natural framework to do this. One framework that I really like is optimal transport. It's again an example of classical mathematics, Gaspard Monge, that was looking at transporting mud from one place to another. For a mathematician, it's moving one probability distribution to another, given a cost function. And it's highly flexible. You can actually, most of the dimension reduction technologies we have today, you can, with some squeezing, fit into optimal transport. And of course, it has been published on. Let me finish by 2 slides, a little bit about the future. So as many of you, I'm of course working heavily with agentic systems, so fleets of agents that help to search the literature, that helps to come up with hypotheses, that helps to write code. And I mean, it's hard to predict, especially the future. But I would say that chess can maybe be a toy model for the future of the human-machine partnership for science. It was game over in 1997 when Kasparov lost to Deep Blue, and he was crushed. I mean, he was very depressed, I think, for some time until he said that, okay, no, we will actually make the human-machine partnership and start to use machines instead. And I mean, today, chess is more popular than ever. And we like to see humans play. And we use the machines to actually say, this is the accuracy, this is the best play. Training and analysis in chess, heavily machine-inspired. And I think that the same thing can go in mathematics. I'm following what is happening with agentic systems together with the lean system, so proof correction system. I think this is the way to go also in science in general, to avoid having false results, invention of things that what is called hallucinations. But these should be corrected with error correction algorithms. And then an important thing are the fair digital research objects. So tracing what is happening, what you as a mathematician did, what the machine did, how you interacted, together with all of the other mathematicians and all these fleets of agents. What was happening? There is now systems that can track the provenance of this. And this, I think, is a key thing for us, both in education and for sure in research. My last slide is clinicians will continue to observe patients and measure. What will change is the depth of what we measure, the depth of the observation. So I'm interested in understanding, if I get sick, I will go to my agent and ask, what do I have? I will show pictures. I will come highly informed to my clinician, and we need to interact. We need to together come up with, where do I fall in this immunity state space? How can I be helped, me as an individual? I want you to make a biopsy and build an organoid model where you can test thousands of drugs so I know what I will respond to. I think this is a little bit the future. And I think these digital research objects in the human-machine partnership will help all of us to connect, also as humans. We will continue to be interested in other humans. And if science comes out or mathematics comes out that we as humans do not grasp, it will not be very interesting for us. So there will be a place for humans, but I'm sure there will also be a place for machines. Thank you so much.
Thank you very much for this very inspiring talk. And I invite the audience to ask questions. If you have questions for Dr. Fontes, please line up and use the microphone.
Just because you brought up the chess analogy at the end, it moved me to wonder about one aspect of this. Now, when you're working on, cancer treatment and so forth, this is so important, just getting technology to get the problem done as quickly as possible, no question about that. And I have this very peripheral understanding what's happening in the chess world in terms of the machine impact. And so I might be wrong by this, but my sense is, right, why did Magnus Carlsen retire from chess? And my understanding was the machine has changed the practice of the game, so people are training themselves on computers, to sort of optimize their openings, and we've lost, or it seems, my sense is you've lost this sort of raw talent, and now it's like how much you work with the machine to get yourself to optimize yourself. And I'm wondering, in the sense that one might say some aspect of the joy of chess may have been lost by the machine revolution. What about the mathematical? So again, we're solving very important problems, but part of science, and especially mathematics, right, for a lot of us is the joy of problem solving. And how do you see the machines affecting that?
Yeah, well, I mean, I'm just a human, but I think of this as the era of the artists. I often say c'est l'entrée des artistes. So we will be artists, we will be creative, because I mean this, and I don't care if there is a machine that can run 100 meters in 1 second. I still enjoy seeing Usain Bolt do it in 9 point whatever he did, right? We are interested in other humans, and this will also be true for creativity and intuition and so on. And there will be no machine in the foreseeable future, no AI, that can actually, let's say, simulate the complexity of a human. We have, for each neuron in our brain, it's connected to around 7,000 other neurons. A transistor in a chip is connected to 3 others. That 2 to the power of 3, that's 8, right? 2 to the power of 7,000 is quite a lot. And this is, I mean, human biology is much more complex. It's not a digital thing, it's an analog, so it's even more complex. And a neuron is connected to a lot of other cells and interacting in many other ways. So there is no foreseeable future that I see where an AI can actually simulate what happens in a human brain. That I'm sure I will be dead before that happens, if ever. It's so much more complex. And I think, I mean, I actually been thinking a lot over the last couple of years, what is the place for us? What is the place for me? And in the beginning, I also was a little bit depressed. And now I feel that, no, I mean, it's liberating. I would be super interested in solving a lot of these problems, also with the help of machines. But we need to translate them to each other, right? We are storytelling species, right? We want to tell stories to each other, whether it's mathematics or whether it's, you know, traveling stories or whatever it is. We like to tell those stories. This will continue. This is my view, but that's a very personal, I mean, I'm just a human.
If not, let's thank Dr. Fontes again, and let me remind...
Thank you.